Hello everyone, this is Jason. Welcome back to another episode of STEM Nerds. In today's video, we're going to be talking about antibiotics. We're going to be looking at how they work and how antibiotic resistance occurs because it's a really important concept to understand. Thanks for being here. When we look at the history of antibiotic discovery, we've talked about in an earlier video, it's one of the most important things that allowed us to extend the average life expectancy of human beings from around 40 years old in 1900 to almost double that 125 years later. And one of the people that was really important in that discovery is this person here. This is Alexander Fleming. He was a medical doctor in World War I, and he got very frustrated because a lot of the injuries that he saw he really couldn't do much about once the injuries became infected. And so he set out after he got out of the military to try to figure out a way to solve that problem. How could he develop something that would prevent infections or cure infections once they established themselves? And so he discovered one day while in his lab, something I'm glad he was paying attention to, because I think if I was in this lab, I probably would not have. He was working with some Petri plates and he noticed that there was a fungus that was inhibiting the growth of microbes, of bacteria. And he recognized right there that there was a chemical there that was anti-life, an antibiotic. Now, he couldn't isolate this. It was beyond his capabilities in the 1920s, but a little while later, in the early 1940s, a couple of scientists, Ernest Chain and Howard Florey, they did isolate this antibiotic. And so what they had isolated was penicillin. And penicillin was given to, oddly enough, somebody who had scratched their nose in a rose garden and got an infection. They gave this person penicillin and it was clearing the infection. The problem was they ran out of the drug before they could completely cure the individual, and he wound up dying anyway. So what was discovered is that penicillin is an antimicrobial. It is produced by fungi, and it's got this thing called a beta-lactam ring. And you're going to see that square structure in the upper right-hand corner with the nitrogen there. And basically, all the antibiotics that have that ilin in the last part of the name have this beta-lactam ring. But what happens is we switch that structure around that's connected to the beta-lactam ring. And when we do that, we give the drug a brand new name. And luckily, it has the ability to impact microbes in a different way. Here's amoxicillin. And so you can see the beta-lactam ring, again, that's square. But the structure that is connected to the square, the beta-lactam ring, is different. Thus, a different name, slightly different mode of action. And we've been doing this now for decades with all these ilins. The problem is, as we're going to see, we're running out of options when it comes to altering a drug that is an antibiotic and still have it be safe and effective in the human body. So one of the things that we have to talk about before we get into the specifics of antibiotics is there are some really important concepts that we have to understand. And one of those important concepts is the idea of selective toxicity. This is huge. Selective toxicity means that a chemical will selectively target a pathogen, but leave us human beings relatively unharmed. And that's what we need from our antibiotics. We need them to target the pathogen, but not harm us, because when we're being harmed, we call those side effects, and some of them can be pretty severe. And so we want a drug that's going to kill microbes, leave us alone. That's selective toxicity. Now, there are ways to measure selective toxicity, and that is called therapeutic index. And essentially what we're doing is we're comparing the amount of drug that it takes to kill a microbe versus the amount of drug that it takes to harm us. And the bigger the difference is, the higher the therapeutic index, the better that drug is at killing microbes and leaving us unharmed. And so these two concepts are linked. Selective toxicity, target microbe, hopefully not harm us too much. Therapeutic index is the mathematical equation. How much room do we have between killing microbe and harming human beings? The other thing that we have to understand is that some drugs are going to be bacteriostatic and some are bactericidal. And what that means is some drugs are going to prevent microbes from growing. Some drugs are going to kill microbes. And it sounds like on the surface that the drugs that are bactericidal are better. And in some ways they are, because they kill the microbes. But we also have to consider everything holistically. Bacteriostatic drugs are probably not going to harm our normal microbiota to the extent that bactericidal drugs will. Bactericidal drugs will often have, when it comes to selective toxicity, they will be less selectively toxic and possibly have more side effects. The next thing to consider is what is the drug's spectrum? And we have two different spectrums. There's narrow spectrum drugs and broad spectrum drugs. Narrow spectrum drugs will target either gram-positive or gram-negative microbes, but not both. Broad spectrum target both. And again, in terms of which one is better, well, it really depends. If you know the causative agent of an infection, sometimes it's better to use a narrow spectrum drug because that narrow spectrum drug will more specifically target the pathogen and hopefully leave a lot of our normal microbiota, our normal flora, unharmed. While the broad spectrum drugs are going to be more useful when you don't know exactly what's causing an infection, and maybe you have a life-threatening disease as well, and that drug is going to wipe out both gram-positive and gram-negative microbes. There could be a lot more collateral damage to our normal flora when using those drugs, however. When a clinician prescribes an antibiotic, sometimes you'll see, hey, you got to take this drug this many times a day for this many days, and that's a really important indication. And those things usually come through in our clinical trials, our phase 1, phase 2, phase 3. And a lot of that has to do with the drug's half-life, how long it takes for half of the drug to break down in the human body. And so if you're taking an antibiotic 2 or 3 times a day, that's telling you the half-life is actually very short. It breaks down a lot faster than a drug you might have to take once a day or once every couple of days. Half-life can be impacted by the person's kidneys, how healthy the kidneys are, how healthy the liver is, and the age of that person. Now, antibiotics will sometimes work well with other antibiotics, or sometimes they will actually work well with other types of medications, and we have words to describe how drugs interact with one another. And so the first one is that the drugs are synergistic. In other words, they work well together. And so when you use these drugs together, they're going to have a better impact, and a really good example of that is trimethoprim and the sulfa drugs. They both target the folic acid pathway. They do it in different ways, but when used together, they're way more effective than when used individually. Drugs can have an additive effect where if you prescribe one and then prescribe a second one, the impact is going to be much greater, but they're not necessarily working together at the same pathway. And then finally, we have the antagonistic effect where one drug is going to limit the effectiveness of another. And so a really good example of that would be, well, if you take an antibiotic and a probiotic at the exact same time, the antibiotic might be undoing a lot of what you're trying to accomplish with a probiotic. There are some antibiotics that will render birth control basically ineffective. And so you have to be really careful when prescribing antibiotics if a patient is taking some other type of medication. We also have to keep in mind that antibiotics can harm human beings. Before we get into the modes of action and what exactly it is they do, keep in mind that antibiotics in some cases can lower immunity. They can be incompatible with anticoagulants. Sometimes they can cause damage to the liver and the kidneys. They can disrupt the normal flora, something we've already talked about. And so if the antibiotics are destroying the normal flora, well, that's going to leave more attachment sites available for potential pathogens. And so we have to be aware of that. And of course, there can be allergic reactions to certain antibiotics. It's one of the first things that a clinician will ask you, especially if there's a suspected infection. Are you allergic to penicillin? Because people who are allergic to penicillin, if that goes into their body, they can have a very, very strong immune reaction. So what is it that antibiotics do? Well, there are different classes of antibiotics, and generally speaking, they're going to work in one of the following ways. Some antibiotics will interrupt protein synthesis of bacteria. Now, when they do that, sometimes what you wind up with is an antibiotic that can also interfere with human protein synthesis as well, because the ribosomes in bacteria look a lot like the ribosomes in the mitochondria, and so there can be some side effects there. When we were talking about broad versus narrow spectrum drugs, a lot of the drugs that are going to inhibit protein synthesis are going to be broad spectrum drugs because both gram-positive and gram-negative microbes must synthesize proteins. Other antibiotics will interfere with nucleic acid synthesis. Again, this is typically a broad spectrum type of drug. And then there are some of the most important and most predominant classes of antibiotics interfere with microbial cell wall synthesis. Now, this is a really nice one because, again, if we go back to some of the concepts that we've already talked about, this can be usually, it's a narrow spectrum drug. It's going to be far more impactful for gram-positive infections, although there are some broad spectrum cell wall inhibitors. But if we're going back to the selective toxicity concept as well, The cell wall inhibitors usually are going to be highly selectively toxic because we human beings don't have cell walls. And so we're targeting a structure that we do not possess. Now, when we look at, again, protein synthesis, nucleic acid synthesis, the selective toxicity might be a little bit less because we do, in fact, have those processes. They're not exactly the same as bacteria. That's why the antibiotics work. But again, when it comes to selective toxicity, the cell wall inhibitors typically are going to have the greatest selective toxicity. Cell membranes, this becomes very, very tricky because, as you know, we have cell membranes, so do bacteria. And so a lot of the drugs that target cell membranes are actually topical solutions, and they're not designed to go into the body. Selective toxicity is a lot lower with these. And then finally, we have the metabolic pathway inhibitors, the folic acid pathway in particular, and we do not rely on the same pathway as bacteria. And so again, when it comes to cell membrane, a lot of those are going to be broad spectrum. Same thing with the folic acid pathway. So here are some really common classes of drugs and what they target. So cell wall inhibitors, again, we've got the illins like penicillin, amoxicillin, cephalosporins, carbapenems, which is a really, really important class of drugs that is more of a second-line drug when the others fail. But these are all targeting cell walls in slightly different ways sometimes. And you got the protein synthesis inhibitors as well. Tetracycline, I think, would be a really important one there, one of the more common. Nucleic acid synthesis drugs. The fluoroquinolones is a really important group of drugs for that particular pathway. Up in the upper right-hand corner, we've got the metabolic pathways, and I've mentioned this already, the sulfonamides and the trimethoprim, those sulfa drugs, those are the ones that are working synergistically to make sure that the folic acid pathway is inhibited. And then you've got the cell membrane disruptors. Daptomycin would be an example. Most of these, once again, are topical solutions not designed to go into the human body. One of the things that we are very concerned with in biology is the concept of antibiotic resistance. These drugs that were developed less than 100 years ago are showing less and less effectiveness when they are prescribed for human beings. And there's a lot of reasons for that. The general way that which that happens is you've got a population of microbes, and there's going to be a diverse population genetically. Usually that's the case. Well, some of the microbes in that diverse population are going to be susceptible to the drug. That's what we want. We want the microbes to be susceptible. However, some microbes may be resistant due to genetic mutation. If the microbes that are resistant are allowed to survive, they begin to repopulate in that environment. And pretty soon, because you have mostly genetically identical microbes coming from those resistant microbes, the entire population becomes resistant. Now, in human beings, we get around that sometimes because the goal really with the antibiotics is to assist our immune system. Our immune system is being overrun. We provide a drug that either stops the microbes from growing, bacteriostatic, or kills the microbes, bactericidal, and that assists the immune system. It helps the immune system out. And then hopefully all we have left are the resistant cells. And the immune system doesn't care if the cells are resistant to antibiotics. It just destroys those cells. Again, if the cells are left behind, that's where the issue is. One thing I want to clear up, we human beings, and it's a language that I don't like when I hear it used in the biomedical fields. And you'll often hear people say, or clinicians say, oh, hey, I'm sorry, we tried this antibiotic. You're resistant to the antibiotics. We got to try something else. Time out. Really important to understand. Human beings need to be resistant to antibiotics. It's the whole concept behind selective toxicity. The whole reason why antibiotics work is we human beings are resistant to the drugs. In other words, we are unharmed by the drugs. It's the microbes that need to be susceptible. And it's also the microbes, when they become resistant, that's the problem. And the reason why that's so important is because a lot of times the bacteria that are associating with the human body can be transient, and they can be transferred from one person to another. And so it's not the person. It's the microbes that are either susceptible or resistant that causes the issue. We human beings should always be resistant. So what do the bacteria do to become resistant? Well, there's a few things. Some microbes can change the proteins on their cell surface and basically block uptake. And so the antibiotics, in order to do their job, have to gain entry to the cells. And if they can't do that, well, the antibiotic isn't going to do a whole lot. The other thing that can happen is sometimes the antibiotics get into the cell, but the cell has a mechanism to inactivate the drug. Maybe something binds to it and stops that from actually having an impact. Antibiotics sometimes get into a cell, but then they are pumped right back out. This is a far more common thing in gram-negative microbes where they have an efflux pump. Again, if the antibiotic can't remain in the cell, well, it's not going to do a whole heck of a lot of good. And of course, there are bacteria that can produce enzymes that break down the antibiotics or alter them in some way. Now, there are two main types of antibiotic resistance. There is intrinsic resistance and acquired resistance. Acquired resistance is the one that we typically think about when we're talking about something like antibiotic resistance. The microbe has acquired new genetic information. Either that's through genetic mutation of its chromosome or the acquisition of antibiotic resistance plasmids. Intrinsic resistance means that the microbe doesn't have the pathway or the structure that the antibiotic targets. I'll give you an example. Let's say you had an individual that had a mycoplasma bacterial infection. So mycoplasma lacks a cell wall. If one were to prescribe a cell wall inhibitor to treat an infection that was caused by mycoplasma, it's completely worthless. And the reason for that is, well, mycoplasma doesn't care if there's a cell wall inhibitor in the environment. It doesn't produce cell walls, and so the drug is going to do nothing. And so that's intrinsic resistance. The microbe does not possess the structure or the pathway that the drug targets. It's acquired resistance that we're typically worried most about because that is due to some type of change in the genome of the microbe. And that change can be passed down from one generation to the next. And so you can have horizontal gene transfer, something we talk about in another video, where a microbe can transfer plasmids from itself to other microbes. That is a hugely important thing that can happen. And then you have vertical gene transfer, where every time a microbe that has a drug-resistant plasmid or some type of a mutation in a chromosome, every time they have offspring, those offspring are going to be genetically identical to the parent, and therefore will also have those mutations that allow for antibiotic resistance. So what can be done to slow antibiotic resistance? The first thing is take the antibiotics as prescribed. And so if you wind up with an infection and the doctor says, hey, I need you to take this twice a day for 7 days, you do that. Why? If you stop the antibiotics early because maybe you just feel better, what you've done is you've probably destroyed a lot of the susceptible microbes, but you may have left behind some of the resistant and some of the intermediately resistant microbes, which could have potentially been killed by the antibiotic, but you stopped treatment too soon. There might be too many microbes left for the immune system to actually clear the infection, and you wind up with more symptoms later caused by a rather serious infection of now mostly resistant microbes. So take the drugs as prescribed. Second thing, once you're done, if for some reason there are antibiotics left over, do not flush those down the toilet. Don't throw them in the garbage. Take them to a disposal facility that can dispose of them properly, whether that's back to the pharmacy. Sometimes municipalities will allow you to bring drugs in so they can properly dispose of them. But if those drugs wind up in the environment, that is going to select for microbes that are going to be antibiotic resistant. The other thing to consider is if you're watching this and you're a citizen of the United States or other developed countries, usually you have to get a prescription for antibiotics. That is not always the case in some developing countries. Sometimes you can just go to the store and get antibiotics just over the counter, and those antibiotics and the concentrations of the active ingredients may be, well, let's just say suspect. And if there's low concentrations of the antibiotic and some of these drugs that should not be over the counter, well, you're just breeding antibiotic resistance. Next one to consider, and this is huge, antibiotics have been shown to have some pro-growth attributes. That's especially important for farm animals. And so originally, antibiotics were put in water systems and in feed for farm animals because a lot of times they are in very close quarters. And if you have one animal getting sick on a farm, it can spread very rapidly. And so it was used as a preventative. But then once it was discovered that the antibiotics have pro-growth aspects as well, farmers unfortunately started using them for that particular aspect and not necessarily to keep the infections at bay or to prevent them. What this means is you wind up with a lot of antibiotic in these feeds, in the water system, and then in their waste, and that winds up as part of the environment, and that breeds antibiotic resistance, and so that absolutely needs to stop. And what's interesting is that countries that have stopped doing that, turns out that their farm animals are just about as healthy, if not more healthy, than the farms in countries that are still using antibiotics as both a preventative and for pro-growth. Next thing is patient education. We need to understand as human beings, antibiotics kill bacteria. They do not destroy viruses and the vast majority of protozoans and fungi. And so if somebody has a viral infection or a protozoal infection, the prescription of antibiotics really isn't going to do much. In fact, often what happens is those things were going to be healed on their own. They were going to be cleared by the immune system. And people think they work because, oh, I'm starting to feel better. Well, it wasn't the antibiotic. It was a function of time. And so prescribing antibiotics only for bacterial infections. Another thing that scientists are contemplating is whether or not certain antibiotics should be pulled off the market when they are deemed, well, let's just say nearly ineffective. And the reason for that is usually the genetic mutations that occur to allow bacteria to become antibiotic resistant, they're not as advantageous as the wild type microbes unless the antibiotic is present in the environment. And that means, what if we took the antibiotic out of the environment and allowed the wild type antibiotic susceptible microbes to outcompete the resistant type? How long would that take? Still unsure. Some scientists say years, so possibly decades, maybe more. In the meantime, we would have to take that antibiotic off the market. All right, everyone, so that is how antibiotics function and a little bit about antibiotic resistance. As always, thank you so much for being here. Have a great week.